this is going to be something completely different. I hope you bear with me for a bit. I'm not going to show any neuroscience models, but I hope you can still take something home from my talk. So I'm going to tell you a little bit about some of the work that I've done towards the end of my PhD at the University of Amsterdam. So now I'm a postdoc here at TNS. But today I'll tell you about this maximally predictive dynamics from data. So as you can see in these videos, nature presents us with complex, multi-scale, unpredictable patterns that defy our understanding. Yet in physics, we've shown a tremendous success at building coarse-grained predictive theories from the principled integration of macroscopic degrees of freedom. So one example of that is, for example, Brownian motion, where by approximating the collisions experienced by a larger particle through Gaussian white noise, we can predict its larger scale diffusive properties. Okay? Now, in order to do this, we generally need either some understanding of the microscopic dynamics or some notion of symmetries or conservation laws that allows us to write down theory. Well, in most cases in data, we don't have this level of intuition. So what we'd like to do is to try to build still these coarse-grained predictive descriptions, but working directly from measurement data. And so in particular, I'm focused on systems from neuroscience, so either behavior or neural dynamics, where we've seen an increase in the amount of large-scale and high-resolution data, but these data are also very difficult to analyze. I mean, how do you make sense of things like that? Like one enticing hypothesis, one enticing idea that we're going to explore is to think about, let's say, behavior or coherent patterns in a neural network as evolving in some complex potential landscape where different wells correspond to different stereotype behaviors, and then fluctuations can drive the system to transition from one well to another. Now, in order to make this analogy precise, we need to be able to infer or extract some slow coarse-grained variables that capture the larger scale properties of the dynamics, such that then the faster dynamics can serve as fluctuations. And so what we're going to do is that we're going to draw inspiration from other fields, namely chemical kinetics, fluid flow, climate sciences, and we're going to use a transfer operator approach to search for such slow observables directly from data. And we're going to do that by studying the time evolution of ensembles or densities in a carefully defined state space. Now, the intuition for this is the following. So imagine you have water flowing down the river. Okay, so on a microscopic scale, the water molecules will just be bouncing around each other erratically on very fast time scales. But if you zoom out and look at a larger body of water, then you'll see that it's actually moving coherently down with the river, okay? And so this is basically what we want to do. We want to, in some sense, integrate out or abstract from this fine-scale randomness and see the larger scale structures, and we can do that by essentially zooming out and looking at the evolution of ensembles. Now, due to the conservation of probabilities, the evolution of densities is linear with time, so you can think about L, for example, as being the Fokker-Planck operator if you have stationary Gaussian random process, okay? And so what this means is that we can, in principle, subsume the generally nonlinear dynamics of the underlying state space and their trajectories into the approximation of a single linear operator for the evolution of densities, okay? So these two descriptions are equivalent to each other, but using this linear operator, then we can decompose the dynamics into a set of modes that relaxes to the steady-state distribution of different timescales that are given by those eigenvalues there. And so the hope is that we can identify from these eigenvalues the fast-decaying modes that represent, let's say, the fluctuations, and then the slow modes that capture the coarse-grained, longer time scale, larger scale properties of the system. Now, in general, when we're working from data, we're not guaranteed to measure all the variables that are relevant to explain the system, right? So we might be missing important degrees of freedom that give rise to complex, non-trivial, non-Markovian effects to the evolution of densities, or might preclude, let's say, metastable states that are living in a higher dimensional space that we don't know because we don't measure those variables, okay? So before we proceed, we kind of have to do some sort of, let's call it inverse physics, and try to learn from our incomplete measurements what is the full set of variables that characterizes the dynamics, which is Markovian and maximally predictive of the future. Okay, so let's look at a physical example for intuition. So imagine you have a particle that it's hopping in a double well potential. So it's going to stay most of its time near each of these wells, and then it's going to hop around. And the dynamics in this case, we call it underdamped, meaning that inertial effects are important, so the dynamics is second order in the position, which means that you need not only the position to predict the future, but you also need to know the velocity, right? So if I just give you the position at some time t, you're not going to be able to tell me whether the particle is going to move to the left, right, up, or down. But if I give you also the direction of the velocity, then you can predict the future. Okay, so these two variables, the position velocity or position momentum, is what we call the phase space variables, okay? And they are Markovian and maximally predictive of the future. Now, let's make an exercise. Let's imagine that we don't know about the physics and we're just measuring the position of the particle. Okay, how can we know that there are these extra degrees of freedom that we're missing? Well, one simple way to get back this velocity information is to just include time delays of our position measurements, right? So given enough time delays, then we can approximate the velocity pretty well. Okay, so this is what we're going to do. We're going to expand our state in time. So we're going, let's say in this case, from a one-dimensional signal to a k-dimensional signal where k is the number of delays that we take. And we're going to try to see, study the predictability of this reconstructed state as we expand in time, until basically we can find the number of time delays that gives us a maximally predictive state, so that we have a correspondence between this delay space and the original full state space. Okay. So it turns out that this transfer operator formalism I just described before, it offers a direct means to the estimation of entropy rates, which is a fundamental measure of unpredictability. Okay, so the entropy rate essentially measures the entropy of the future states given the current state. Okay, so in some sense it tells you how broad are the possibilities of the future depending on where you are right now. Okay, and in practice, when we approximate this transfer operator, what we do is that we make tiny partitions in the phase space, okay, and then we count how many transitions occur between these different partitions, and we build a transition matrix. And you can see here that from that transition matrix, we can estimate directly the entropy rate. So here, Pij is one of those matrix elements, a transition matrix. And pi of I is the corresponding stationary distribution that you can obtain just by taking its first eigenvector, okay? So you can see that you can compute this entropy rate depending on how many delays you take, so how much you expand your state, and depending on how many partitions you take, so how finely you discretize your state. And generally, what we observe is that as you include these time delays into your state, you essentially narrow down your prediction of the future, okay? So you're using past information to narrow down your prediction of the future. And if the system is Markovian on some time scale, then you should be able to see that after some amount of delays, you don't do any better at predicting the future. So you don't need to bother more about the past. And that's kind of what you see here in this plot, where I'm showing you the entropy rate as a function of how finely I discretize the state space for different values of this number of delays, okay? And you can see here that clearly just measuring the position, it's going to be very unpredictable. So this is k equals 1. But as soon as I start adding time delays, then you see that these curves will collapse. And at a certain k, basically, I've reconstructed the state space. I get this maximally predictive state, which we hope maps to a more Markovian description of the system. Okay, so now that we have this reconstructed state, we can build our transfer operator and look at its spectrum. And as expected in this case, then the slowest eigenvalues will correspond to the slow hopping between the two potential wells, okay? And in fact, we can predict the hopping rate directly by just looking at those eigenvalues. And I'm showing you here that as a function of temperature. You can think of that as noise intensity. And the corresponding eigenfunction, so the eigenfunction that corresponds to these eigenvalues, it identifies these two wells, these two metastable states. You can see that the sign structure splits along the energy barrier, and negative values are on the right side and positive values are on the left side. Okay, so you can use these eigenfunctions to identify these coarse-grained metastable states. And this has been done in many fields, and what I'm going to show you now is how this can be applied also to a system that it's more relevant for neuroscience. And in particular, we're going to look at the naturalistic behavior of the C. elegans nematode. So C. elegans has been a model organism in biology for a very long time. We know a lot about its connectome, its genes, and we're interested in trying to understand how behavior is modulated on different time scales and length scales by this underlying genetic and neural machinery. Okay, so in a nutshell, the worms move by propagating waves along their body, which moves them forward, backwards, or turning in seemingly random sequences. And it is by sort of chaining these sequences or these movements in time that the worms change the way in which they navigate and explore an environment. Okay, so here, this data is taken from some collaborators of ours in the University of Toronto, Will Ryu. And this little red box there on the right is the microscope that is following the worms around on the plate so that we get very high resolution images of the worm. Okay, so already here you can see that behavior, even at the level of just a nematode with 300 neurons, is very multi-scale in nature, right? So you can think about behavior at the scale of the fine-scale muscular activation by the neuromuscular junctions, or you can think about behavior at the scale of like navigation, feeding, mating, or even longer things like aging, right? So it's very multi-scale in nature. And so in some ways, it's analogous to trying to find these metastable states or rare transitions in a complex potential landscape. So let's see how this works for these worms. So first of all, we choose to study the behavior from a worm-centric point of view. So we remove the translational and rotational degrees of freedom, meaning we don't really care if the worm is moving forward to the left or forward to the right. We still want to call that moving forward, okay? And so basically, we parameterize the shape of the worm. And it turns out that at each frame, the shape of the worm is pretty well characterized by a linear combination of 5 primitive shapes. So basically, by changing these 5 numbers here, I can generate any type of shape that the worm can do. And so this means that I can translate a video like this into a time series like that, okay? A 5-dimensional time series that now I can use to do my analysis like I've done before. And so then we do the same that we've done for this double well potential. We add time delays to these measurements of the worm. And we see that after roughly about 0.5 seconds that these curves essentially collapse and that we have reconstructed the state space, okay? So the state of the worm now is composed of short posture sequences, short movements, short little waves, okay? And now given this transfer operator, we can do our spectral decomposition, and we actually find slow dynamics that evolve over time scales of order 10 seconds, so almost 2 orders of magnitude longer than the fine scale dynamics. And interestingly, these correspond to long time scale behavioral states that have been identified that C. elegans uses for navigation. Okay, so here I'm showing you a projection of the corresponding slow eigenfunction onto the space spanned by the body wave phase velocity, which captures the frequency of the worm moving forwards or backwards, and the curvature of the body, okay? And you can see that on the one hand, this red patch here, negative values, correspond to the worm basically just moving forward with very little curvature, and in blue, so these positive values correspond to the worms moving backwards and then turning. And this is a maneuver that it's been coined a pirouette, and these 2 states are what the worm uses for navigation. So here you can see that clearly at the trajectory level. So this is the trajectory of the centroid of the worm, so where it's going. And you can see that in red, basically you have these runs that are interspersed by these pirouette events. So it's very similar to the kind of run and tumble description that we get for E. coli, but here it's for a multicellular organism with a brain. And remember also that I'm not using any information about its position, okay? I'm only using the posture itself to identify these states. So it's kind of surprising that you can get back these larger scale things. So if we now subdivide the dynamics into these 2 coarse-grained states, we find essentially 2 time scales, a longer one that extends to hundreds of seconds that corresponds to these long runs, and a shorter one that corresponds to these pirouettes, okay? So we get a way to do this principal coarse graining of behavior at this scale. But it turns out that the transfer operator is also a very simple but very accurate model of the dynamics, okay? So I can go now, take my transfer operator and just sample states according to the rules given by my transition matrix, okay? So essentially, I'm going to be diffusing in some high-dimensional state space, and we can map that to the postures. And it turns out that the simulations are very accurate at the level of the microscopic posture dynamics. So here I'm showing you the body curvature as a function of time for data and simulations, and on the right you have the corresponding kind of animated worms. You can already kind of see that the dynamics resembles between the simulation and the data. And on the other hand, besides capturing these finer scale dynamics, we can also capture longer time scale dynamics. So we can now put these worms on a plate and we can make them move, okay? And so what I'm showing you here is the position of the worm in the lab frame. So this is on a much longer time scale. We're talking about minutes now. So red and blue are, again, these 2 different states. And so we can generate these trajectories, and it turns out that these trajectories actually resemble really well what we observe from data. In fact, we can measure the mean square displacement for these trajectories and compare it between data and simulations. And we see that our simulations capture the full transition between ballistic motion and diffusive motion at larger timescales. So gray is the simulations and the dashed black line is the data. And so we can even infer a diffusion constant, an effective diffusion constant in this diffusive regime. And we see that our model and the data match pretty well. So here just green is just a shuffle where we destroy the dynamics but keep the relative abundance of runs and pirouettes, and that makes a huge offset to the diffusion constant. But if we keep the full model, then we can predict this diffusion constant. And again, this is a very non-trivial jump in scales, okay, that it's obtained from a very simple model. It's just a Markov chain, but built from this reconstructed state space. So by thinking principally about the representation, we can simplify our theory and still be able to make pretty dramatic predictions and bridging timescales in behavior. So we're now interested in trying to understand, what kind of neural and genetic perturbations we can do to modify the behavior at these scales. Because we can study behavior, as I showed you, from the microscopic fine-scale postural movements all the way up to the way it navigates and explores the environment. And I'm also interested if you have any ideas of examples from neuroscience or data where this kind of approach would be useful. I'd be happy to hear your thoughts on that. And I'll just leave you with our recent archive preprint, and I'd like to thank my collaborators and thank you for listening. Thank you.
Thanks a lot. Comments or questions?
Thanks for a great talk. I would like to ask you, how well does your transfer operator represent more than one C elegans, let's say, and also whether there is a way in which you can incorporate sudden changes in macroscopic behavior. Let's say you have an animal that at some point decides to do something non-trivial, and so what happens to your transfer operator then?
So like this whole transfer operator description kind of assumes, in some ways, from the way that I'm doing it, that I'm in some kind of ergodicity limit, okay? That I've observed the dynamics for a long enough time such that I can capture its longer time scale properties. So whatever I'm inferring, it depends on the time scale of my measurements, right? So if I'm looking at a much longer time scale measurements where the worms actually do more non-trivial things on longer time scales, then I can pick up on those. But in this data set, it's just a foraging data set where the worms stay on the plate for half an hour, and these are the longer time scale things that happen. In fact, there is a slow transient to the dynamics that comes from the habituation on the plate that we cannot pick up from the transfer operator, okay? Because it's so slow. It's slow, like much slower than the measurement timescale, that I cannot really pick up on it. So essentially, you will find whatever that is in your data set. So yeah, if you have those types of things happening in your data set and you can sample them long enough, then you will be able to see them. Otherwise, no. Yeah. Another thing you can do is that you can sample across multiple worms, multiple experiments, and you can think about each worm as a different initial condition for your dynamical system, in which case then you will be able to see these longer time scale properties. Yeah, good question.
Yeah.
Thanks, that was really interesting. I was wondering how you're choosing the partition of the state space to come up with the transfer operator.
Okay, so that's an interesting point. So the partition, I do this maximally, I try to maximize the entropy of the partition. So given my data set, I devise some Voronoi cells, okay? And I try to make them fine enough to maximize the entropy, but not too fine so that I start having finite size effects. Okay, so there is a trade-off between how much data you have and how finely you can discretize your state. But what I try to do is to capture as much information as I can in my partition before I do this subdivisioning again. Yeah.
Thanks for the nice talk. I was just wondering, could you see collective behavior of your worms in this model?
Yeah, so that's a good point. We haven't tried to do that right now, but you could think about applying the same type of formalism to collectives of animals. And there is now data on this in worms, and actually worms do crazy collective behaviors. But we haven't done that yet. And to be honest, I'd have to think about how this would generalize to a collective. It is non-trivial due to the properties of how the densities will evolve. But it's a really good, interesting possibility that we'd like to explore. Yeah.
Okay, other question?
Oh.
We did do this in collectives of neurons, though, which is kind of similar in the worm. And you also find the same types of long time scale neural states. So collective neural dynamics on long time scales. Yeah.
Thank you very much for your talk. I will have a very naive question and try also to connect to neurobiology. I was wondering, do you know, in terms of ethology, why these worms are doing this oscillation? Is it protective? Is it because it's the best way to track on your way any source of feeding? Or
that's an interesting question. So we haven't actually analyzed whether, I think what you're asking is whether these kind of macroscopic states are optimal for like dispersal and foraging, or to trying to find food sources, or
because for me, I always try to do connection, but so it really looks like an oscillation. And I was thinking, okay, maybe we could use this type of oscillation to reproduce that we have in the brain. And I was thinking,
I see.
Imagine that you have something like I don't know, which arrive in your petri dish, like a wave of water or anything else. Could we simulate? I don't know, like something that happened in the brain and could affect this oscillation.
Yeah, you can.
I was thinking about a model like for disease in brain, like when you have an AVC and a stroke, and so you have blood arriving, just what's happening on the oscillation of the brain.
Yes. So that's a really good question. I mean, there's been some work on this on more mechanistic models of the C. elegans brain. So the C. elegans neurons are a bit weird in the sense that most of them don't spike. They have a more kind of continuous activity. But some of them have been kind of identified as central pattern generators. So they keep some sort of frequency. And they keep a coupling between the head and the tail. And people have looked at different types of perturbations they can do on different neurons and how that impacts the behavior. But so, I mean, we are now starting to look at different types of perturbations, both genetic and neural perturbations, so either ablation experiments or mutation experiments. And we're seeing like very interesting changes in the behavior that come from, let's say, if I ablate a specific type of neuromodulator receptor, that's going to impact the behavior on a very long time scale. So we are starting to ask more kind of mechanistic questions about the underlying control mechanism. Yes, it's a really good question.
Yeah, thank you for the talk. I just have a technical question about how you expand the state with delays, how you choose this. I was not sure.
So how do I expand the state with delays? So I basically just concatenate these delays in time. Okay, so I don't know if I have a. So let's say you have a time series that goes from x1 to xt. Okay, then for example, for k equals 2, I build a matrix like this. Okay, to xt minus 1. Just from a practical perspective, this is what you do. You expand the state, but you add a dimension that it's a temporal dimension. And actually, I mean, this type of approach has been, proved by mathematicians a long time ago to actually give you a precise mapping between this delay embedded space and the underlying phase space or the state space of the dynamical system. So that's the so-called Takens delay embedding theorem. Takens wrote it for deterministic systems that it's been expanded now to recently in the 2000s by Stark to general classes of stochastic dynamics. So you can basically build a mapping between the original, let's say, n-dimensional phase space and the delay embedded phase space such that 2 points are like diffeomorphic to each other, meaning like they are the same, and their neighborhoods are the same. So if you study the dynamics on one space or the other, you should be able to find the same properties. They preserve all the ergodic properties of the system. Yeah.
Thank you.
Okay, thank you.